So my section starts on dysfunctional androgen receptors can block male masculinization, and it starts on androgen insensitivity syndrome, which is a syndrome caused by a mutation of the androgen receptor gene that renders tissues insensitive to androgenic hormones like testosterone. This affects XY individuals that are phenotypic females, but they still have internal testes and regressed internal genital structures. The gene for androgen receptors are found on the X chromosome. So if the gene is dysfunctional in XY individuals, they'll be incapable of producing normal androgen receptors, and their tissues will fail to respond to androgens. These people have normal SRY, so they develop testes that secrete AMH and testosterone. But in the absence of working androgen receptors, the Wolffian ducts fail to sense the testosterone and thus regress. So the external genital tissue forms labia and a clitoris. Women with AIS develop breasts, but they do not menstruate and are infertile. The next section is some people seem to change sex at puberty. So babies are occasionally born with a genetic mutation that disables the enzyme 5-alpha reductase that converts testosterone to DHT. These are XY individuals with a condition which they develop testes and normal male internal reproductive structures, but their external genitalia will not masculinize fully until puberty. Their genital epithelia, which normally possesses 5-alpha reductase, is unable to amplify the androgenic signal by converting the testosterone to more active DHT. So the phallus is only slightly masculinized and resembles a large clitoris. The genital folds resemble labia, although they do contain testes. At puberty, testes increases androgen production and the external genitalia become more fully masculinized. Their phallus grows into a small but recognizable penis, and the body develops narrow hips and a muscular build without breast, and the individuals begin to act like young men. This syndrome is known as Guevadoches, which is literally eggs at 12, and it's a nickname for individuals who are raised as girls but at puberty change appearance and begin behaving as boys. The next section is how should we define gender by genes, gonads, genitals, or the brain? Behavior is much more difficult to define as feminine or masculine. Only behaviors displayed exclusively by one sex is at childbirth. XY AIS identifying as females is a good example of this, as they are genotypically males but phenotypically females, and they identify that way. The same hormones that masculinize genitalia of the male animals in utero also masculinize the brain to affect later behavior. So that's the next section, gonadal hormone direct sexual differentiation of the brain and behaviors. So researchers were able to induce lordosis with progesterone injections in guinea pigs. Female guinea pigs that were exposed to testosterone in utero did not show lordosis. This can be an example of the organizational effect, which is a permanent alteration of the nervous system, and thus permanent changes in behavior resulting from the action of steroid hormones on an animal early in its development. The steroid has an organizational effect only when present during a specific sensitive period. The sensitive period is the period during which in development in which the organism can be permanently altered by a particular experience or treatment. For rats, androgens given just after birth can affect later behavior. Guinea pigs, however, must be exposed to androgens before birth for adult lordosis behavior to be affected. In mammals, puberty can be viewed as the second sensitive period. The next section is early testicular secretion results in masculine behavior in adulthood. The organizational hypothesis provides a unitary explanation for sexual differentiation. A single steroid signal, usually an androgen, diffuses through all the tissues, masculinizing the body, brain, and behaviors. From this point of view, nervous system is just another tissue that's taking note from the androgens. If the nervous system does not detect androgens, it will organize itself in a mostly feminine fashion. Full masculinization behavior requires androgens both in development and in adulthood. The next section is the estrogenic metabolites of testosterone masculinize the nervous system and behaviors of rodents. So there's some paradoxical findings in this. The first was that newborn females treated with estrogen failed to show lordosis behavior in adulthood. In fact, if these females were treated with testosterone in adulthood, they would show mounting behavior, indicating that these early estrogens had masculinized their brains. But the question that people had was, why would estradiol cause masculinization of the brain sexual circuitry in females? So the answer comes down to that estradiol is a principal metabolite of testosterone. In a single reaction called aromatization, the enzyme converts testosterone to estradiol. Aromatization is a chemical reaction that converts testosterone to estradiol and other androgens to other estrogens. And aromatase is an enzyme that converts some androgens to estrogens. The ovaries normally contain a great deal of aromatase, but generally so does the brain. So this leads to the aromatization hypothesis, which is a hypothesis that testicular androgens enter the brain and are converted there into estrogens to masculinize the developing nervous system in some rodents. This is done through alpha-fetoprotein, which is a protein found in the plasma of fetuses that binds estrogens and prevents them from entering the brain. Male rodents' brains are masculinized when testosterone bypasses alpha-fetoprotein and enters his brain, where it is aromatized to estradiol within individual neurons. If the male rat is castrated at birth, it grows up to have a small penis and shows fewer intermissions, even when given replacement testosterone in adulthood. Castration plus androgen DHT, which cannot be aromatized into estrogen, results in a male with normal genitalia but little or no masculine sexual behavior because of the lack of organizational estrogenic stimulation. Conversely, male rats castrated and treated with estrogens as newborns mount and show intermission behavior when treated with testosterone in adulthood, despite having a very small penis, due to the lack of DHT. In primates, aromatization cannot play an important role in masculinization of the nervous system. The human brain produces a lot of aromatase, but men who have mutations in aromatase genes still have masculine gender identities and sexual development. Men with defective estrogen receptors also develop masculine gender attributes, so hormonal masculinization must be accomplished through androgen receptors rather than through estrogen receptors in primates. The next is box 12.1, which is a paradoxical sexual differentiation of the spotted hyena. So, previously, researchers thought hyenas were hermaphrodites, which is an individual that possesses the reproductive organs of both sexes, which are either simultaneously or at different points in time. The reason they thought this was because female hyenas have a clitoris that is large enough to be, or as large as the penis of a male hyena. Females are also larger, more aggressive, and more socially dominant than males. In other mammals, placenta rapidly aromatizes androgens into estrogens. This conversion may be a way to protect the mother and fetal females from androgens produced by fetal males. But the placenta of the spotted hyena is deficient in aromatase enzymes, so all hyena fetuses are exposed to high concentrations of androgens, which accounts for their masculine appearance and behaviors. With this said, females still mate with males, although they are reluctant to be in close proximity in other situations. This next section is several regions of the nervous system differ between males and females. Difference results primarily from androgenic masculinization of the developing brain. Sex differences in number of synapses were identified in the preoptic area of the hypothalamus, but scientists found that there are many more differences in brains, including differences in neuronal numbers, sizes, and shapes. Sexual dimorphism is a condition in which males and females show pronounced sexual differences in appearance. Song control regions in male songbirds. Male canaries and zebra finches produce elaborate songs to attract mates, but females don't. The brain nuclei controlling these songs are 5 to 6 times larger in males. One of these major nuclei are the robustus archistriatum, also known as the RA. This controls motor neurons innervating muscular singing organ called the syrinx, which is the vocal organ in birds. The RA is in turn controlled by the higher vocal center. Exposing female hatchling female zebrafinch to either testosterone or estradiol causes the development of a larger male like HVC and RA. Female zebrafinches treated in adulthood do not sing well. Singing canaries requires only adult androgen treatment. This has a lot to do with their mating cycle. Canaries mate all year, while zebrafinch only mate in certain time periods, so that changes how their bodies react to certain hormones. The next section is the preoptic area of rats. Sexually dimorphic nucleus of the POA, also known as the SDNPOA, is a region of the preoptic area that is 5 to 6 times larger in volume in male rats than in females. Males castrated at birth had much smaller SNDPOAs in adulthood, while females androgenized at birth had large male-like SDNPOAs as adults. Castrating male rats in adulthood did not alter their size of the SDNPOA. So rat SDNPOA conforms to the aromatization hypothesis. Testosterone is converted to estrogen in the brain and binds to estrogen receptors to masculinize the nucleus. XY rats that are androgen insensitive have testes but have female exteriors. These rats have masculine SDNPOAs because their estrogen receptors are normal. Androgen insensitive rats do not display lordosis in response to estrogens and progesterone because testosterone that's secreted early in life is converted to an estrogen in the brain and masculinized their behavior.